All right, welcome back, fellow scientists. So hopefully you have already taken your Microsoft Forms Lymph System 2 quiz. If not, go back and do that because that's a timed thing. I think I set it to go on at 11:30, and so it'll be open until 12. Open note, open book. So all of you should score 100% on that. So, anyways, do that. And then thinking ahead to the rest of this week and then into next week. Today we're going to finish up the immune system. Thursday I will. So today's just a video, and then there will be a little bit of reading that you'll have to do after you watch the video. And then Thursday I'm going to have a review guide, and I'll give you time, our class time, to work on that review guide. Monday we'll have a Zoom class meeting and we'll go over the review guide and I'll answer any questions. Then I think I'll set up a Kahoot. I don't think we'll try to do it live because that didn't really work too well. I received feedback that people didn't like that. So I think I'll try to set it up so that it's kind of like Microsoft Forms where it's a timed quiz, but you're all doing it kind of at the same time. So we'll experiment with that. I don't know exactly how that works, but we'll see. So that'll be Monday, and then next week Tuesday we'll have a quiz on blood, lymph, and immune. So I think that's chapter 10 and chapter 12. I think chapter 10 was blood, and then chapter 12 is lymph and immune. So, anyways, that's kind of the schedule going into next week, and then next week Thursday, then we'll actually start the endocrine system, which is actually going to be the endocrine and reproductive system is going to be our last unit of the year. So anyways, let's go ahead and let's just dive right in. So we talked about in the last video about how there's two different sides of the adaptive immune system, right? There's the humoral side and the cellular side. Oh, by the way, Shaylin asked for forest green or hunter green. Dun, dun, dun. So there we go. Love it. All right, so this is the humoral side of the adaptive immune system. So basically, it's pretty straightforward. We have these B cells. Remember, why are they called B cells? That's right, because they mature in the bone marrow. So we have these B cells that are just kind of floating around, right? They're genetically made. They're genetically determined. We have the genes to make them. They're also passed from mom to baby in blood and in breast milk. So we have these just kind of floating around, just kind of circulating around in our bloodstream. And then each B cell, you'll remember, has one specific antibody. So you'll notice that this cell doesn't have like this green one or this it's blue one right here, but each has a specific antibody that reacts to a specific antigen. Right? Remember our mantra from biology and AP biology: shape determines function. Okay, okay, perfect. So when this antibody interacts with this antigen, then that stimulates the cell to divide, and it's called clonal division because it clones itself basically, and it divides like crazy, and it'll make two kinds of cells. It'll make the plasma cells. The plasma cells, their only job is to just crank out antibodies, crank out antibodies, crank out antibodies, like thousands and thousands and millions of these antibodies that are specific, come on, to this antigen right here. They call it antigenic determinant, whatever. It could be a molecule, it could be a cell surface protein, doesn't matter. It all means the same thing. So they crank out antibodies. So that's most of the cells that are made from that first original B cell. But then also we have here memory cells. Now these memory cells, these last for up to 10 years. So these last for up to 10 years, and this is what gives your immune system basically memory in essence. So we used to have a small population of these B cells for a specific antigenic determinant. But if our immune system learns that, oh, hey, this is something that we might encounter in the future, we live in an area where this is endemic, then they'll make these memory cells, and then those memory cells they'll make it so that the immune response next time is greater, and we'll talk about that with the next slide because there's a couple things that plays into. All right, so these antibodies, now we have tons and tons and tons of antibodies specific for this antigen floating around in our blood. So what do? This would be a good thing to memorize, right? What do antibodies help to do? Well, they help to neutralize it, right? So if a virus is surrounded by antibodies, it's like a protective coat, and it's not going to be able to infect the cell. And it also, as you can see here by the little diagram, it enhances macrophage activity, or it enhances phagocytosis, endocytosis, whatever you want to call it, if it's surrounded by antibodies. Agglutination, right? If there's actual cells, if the antigen is an actual cell, it kind of clumps all those cells together and makes it easier for these phagocytes or these macrophages to find, right? Precipitation. Now, you chemistry nerds, you know that if you're not part of the solution, you're part of the precipitate, right? So precipitation is something that kind of comes out of solution. So if they're soluble antigens, things that are dissolved in the blood, then they get bound to the antibodies, and the antibodies, like, they make them appear. Basically, they make them come out of the solution and be more visible and more findable again by these macrophages and yeah, by these phagocytes. And then last but not least, we talked already about complement proteins. Let's see, where is it? Right here. So we talked already about complement proteins as being kind of the one of the general defenses. This could also be one of the more specific defenses, right? So if we have our antibodies, right, attached, and then this the complement protein attached to the antibodies, and then makes this pore, and then the water flows in because water wants to go from an area of higher concentration to an area of lower concentration. There's a lot of solutes inside the cell. Water follows salt, right? All that sort of stuff that we remember from regular biology. So that would be what is it? Fourth thing that antibodies do to help protect us. So that's basically the overview of the humoral immune system. Now, one thing I found this interesting graph, both in your book and online. Your book has a slightly different version, but it's pretty much the same thing. So we have a certain population of native B cells, and if we never encounter an antigen specific for that B cell, it's just kind of there, it just kind of floats around. We kind of make more of it as they die because we have the genes to do it, but it doesn't really get activated. Now, if it gets activated, then it's going to stimulate the plasma cells and the memory cells, but there's going to be more memory cells than there were regular cells before it encountered the antigen. And so then upon a second exposure to that specific antigen, then the immune response is going to be much greater, right? Sometimes that's a good thing, sometimes that's a bad thing. That's a bad thing with allergies, right? So that's why sometimes if you have allergies, right, you can develop allergies. Your body can learn to have allergies over the course of its life. And sometimes, like if you get a bee sting, right? Classic example, you don't know if you're allergic to bee stings until the second time you get stung, because the first time you get stung, your body doesn't mount a huge immune response to it. It's kind of learning what this is. And then the second time that you get stung, you have your bee memory cells here for the stuff that's in a bee sting, for the toxins or the venom or whatnot that's in a bee sting. So you have your memory cells, so that's going to create a bigger immune response, and so that's why you only find out if you're allergic with your second dose. At least that's how it's been explained to me. Some of you have physicians for dads and moms, and they can probably explain it better. So that's one thing. Second thing is that this is why these memory cells right here. This is why vaccines work. All right. Now, as a science teacher, I shouldn't have to say this, but I feel like I do. I just want to make it totally 100% perfectly clear. Vaccines do not cause autism. Vaccines are good, right? There was the whole vaccine autism link comes from one study that was poorly done and has been discredited like to an infinite degree. And the guy that did it has been like disbarred from the medical association and all sorts of stuff like that. But yet it persists, unfortunately, because people just don't have scientific minds, and they don't understand yeah, all the stuff that goes into it. So a vaccine is a weak, or the scientific word is attenuated version of a virus, Vaccines, by the way, only work for viruses, right? You can't get a vaccine against a bacteria. Okay, so it's a weak or attenuated virus that you introduce into your bloodstream, and then basically you're teaching your native B cells what it looks like, right? I think I said in regular biology, a vaccine is kind of like a wanted poster. It's like, hey, this is what the bad guy looks like. Learn what he looks like, so that if you see him, you can take him out, right? Because yeah, our cells are all bounty hunters. There you go. So we're making so first encounter, right? That's another thing that people. Oh, I don't want to get the flu shot because like that gives you the flu because then I get a headache and runny nose and blah 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 blah. Well, yeah, okay, it gives you a little bit of the flu, right? And because it's a weak, it's attenuated virus, it gives you like I don't know, 25% of the flu, right? You feel a little bit down, you feel a little bit bad, but then your immune system kicks it into high gear and it's great. But the thing is that it makes these memory B cells, right? So then these memory cells, the next time you actually encounter the flu, right? Like out in real life, like the full, like no kidding, like I'm laid out for a week because I just feel miserable, right? You're not going to feel miserable because your memory B cells, they know what it looks like, and they're like, boom, I'm going to kick this into high gear, and we are going to take care of this flu virus right now. And so, anyways, vaccines are good. Please, please do not be one of those people that is anti-vaxxer. Anyways, I digress. All right, so that's the humoral immune system. So now let's go ahead and let's look at the cellular arm of the adaptive immune system. So cellular immunity is called this because remember we got to go back to our cows. Sometimes with some sicknesses, right? You could transfer the plasma and that worked, but sometimes you had to transfer the actual cells. And that was the only thing that kept the healthy cow healthy. So it's called cellular immunity because you have to transfer the white blood cells. Remember, the antibodies are just kind of free floating in the plasma in the liquid part of the blood, and humoral means liquid. Yeah, all this ties together. It's absolutely amazing. So cellular mediated, it starts with these macrophages, or we could also say an antigen presenting cell because there are other antigen presenting cells besides macrophages. We won't get into that right now, but basically we'll just say this is a macrophage. Right? And so it digests, right? You remember our riders of Rohan and our Urukai, right? So it digests the bad guy and then it presents the bad guy, the part of the bad guy on the surface of itself. So that's going to activate these little T helper cells. So this is a T helper, right? And remember, they're T cells because they mature in the thymus, perfect, good job. So T helper cells, they basically do two things. So here's our little macrophage right here, right, and then here's our T helper cell, right here. You guys can read. I don't know why I'm drawing arrows, but I am. So T helper cells, they actually help with the humoral immune system. So they're going to stimulate B cells, and B cells are just going to go nuts and just like divide, 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 antibodies, 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 right? But then they also stimulate or activate these T killer cells. So they're still T cells, right? They still mature in the thymus, but they have a different job. They don't activate B cells. They don't interact with the macrophages really. They will go and they will find an infected cell, so infected with a virus, infected with cancer, something like that, and they'll attach to it and they'll release molecules, they'll release enzymes that will make these little pore complexes that will cause the cell to lyse, that will cause this infected cell to be destroyed, and so then they detach and then they go and they look for other infected cells. Yeah, and so this is a little bit more specific. Your book talks about how let's see, how the humoral immune system, it just needs to interact with the bad guy, right? And then the cellular immune system, the T killer cells, it needs to interact with the bad guy, and it needs to know that the bad guy is inside a good guy's cell, basically. So your book calls it kind of like a double handshake, like a double check, or yeah, you have to have the bad guy present, and you have to like know that the bad guy is inside of a good guy cell. Um, so anyway, so that's the cellular mediated. We could say a lot more. There are whole entire courses on med school, and like you could become an MD in like immunology, and those guys, yeah, crazy smart, more degrees than Fahrenheit. But anyways, we're not doing that. We're just high school anatomy and physiology students. So I just want you to know humoral, cellular, and just the basics of it. So we have this last graphic right here, kind of summarizing the two, right? So I like this because antigen can stimulate B cells directly, right? Antigen has to be engulfed by a macrophage or an antigen presenting cell before it can stimulate the T helper cell, which stimulates the T killer cell, right? And then I kind of wish that with this diagram, they would put kind of like this, right, up here, because this looks like this is only happening after the second exposure, but it's not. It's happening like during the first exposure, right? And then here, right, would go up there as well. And so then we have our memory cells, and then we have our second exposure, and then that's gonna make that. And really, like this B cell to make plasma cells, that's part of the first exposure, right? Because this antigen second exposure is on this line. It's not on this line. So, anyways, I just think they could have made it a little more, I don't know, a little bit more intuitive, right? If this is time, then it's first exposure, second exposure, and this looks like it's below time. Anyways, maybe I'm reading too much in the diagram. Maybe you understand it perfectly. But just know that first exposure, right? Antibodies are made, memory cells are made, and then the second exposure, the response is enhanced or kicked up or whatnot. All right, so that's it for the presentation. I do want you to dive into your book a little bit. So hang on one second. So this is chapter 12, page 429, right? And your book talks about a whole bunch of stuff that can go wrong with the immune system. So autoimmune diseases, right? It also talks a lot about allergies. It has like two columns on allergies and stuff like that. I don't really feel that it's necessary for me to go through and to read all that. So I would like you, once this video is done, to go to your book and to just read through that, familiarize yourself with that. If you want to, just jot down some really quick notes. But that will be some information that I will ask you about on the review guide and on the test. So that's it for me. That's it for this chapter, Blood, Lymph, and Immune. Review guide on Thursday, review day on Monday, and then test next week, Tuesday. Hopefully, it has been fun and informative. And like always, I will see you on the flip side.